so my name is Asja Radja, and I'm currently in my position as an NSF Simons Quantitative Biology Fellow. And as Moore mentioned, I'm interested in pattern formation in various biological systems. So today specifically, I'll be telling you about patterns on pollen. And just to give you a sneak peek of what the answer is of how these patterns form, they form through a polysaccharide phase transition process. And I'll explain to you exactly what that means and what each of those terms mean. To get started, my general career goal, just so you get a sense of who I am and how I do the work that I do, is to identify quote unquote rules of life. And what I mean by this is I want to understand what are the physical principles that biological systems harness to create a variety of morphologies. So I look at, as I said, different biological systems. So the three that I'm currently studying are the three that are shown here: pollen grains, and then these are actually soft coral sea fans on the top right. And on the bottom, you have a picture of pavement cells, which are single cells that are found on leaf surfaces. And I use a variety of different experimental techniques that are simple for someone like me that are able to do them. And then furthermore, I try to actually quantify the processes that form these different morphologies using physics and math. Okay, so the way that I do this more specifically is I'm trying to essentially see and understand if we can predict form from the genetic code. So if we assume that the picture is you go from a DNA molecule that has a lot of encoded information and you somehow eventually get to the final form of patterns, the question is, how do you do this? And we don't actually know how to do this in a lot of different systems, but you can break up the process into two different steps. So the first step is DNA is going to produce a variety of macromolecules. So you can get proteins, and then after that lipids or complex carbohydrates. And then once it produces these macromolecules, the macromolecules themselves will interact and somehow self-assemble into the final forms that we see. Of course, not all systems follow this exact paradigm, but I look at systems that do, and you can sort of cleanly separate these two processes. And I'm not interested in the first portion, that is other people's work that's very important. I am interested in the second portion, which is this self-assembly process. How in the world do you go from these raw materials of macromolecules to a final form of the biological system in a way that is completely independent of the genetic code? Okay, so that's basically how I approach all of my systems and all of my problems. And I would like to prove to you in this next slide with a little sort of interactive game that this truly is possible in a lot of systems, in the sense that you really can get self-assembly processes that are completely independent of the genetic code, because they also happen in a variety of physical systems that have nothing to do with biology. So here I have 4 different pictures of 4 different patterns. And 2 of these patterns are from purely physical systems. So you can think of like clouds in the sky or waves in the ocean. And the other 2 are pictures of biological systems that have patterns. So either in the chat, or if you want to unmute yourself and take a guess, does anybody want to gander at which of these 2 is a purely physical system and which is a biological system? And Moore can't answer because she's seen this before. I'll give everyone a couple seconds to decide whether or not they want to speak up.
Sorry, exactly how do you define the distinction between a physical system and a biological system?
Thank you for asking the question. So I would argue that waves on an ocean are a purely physical system. There's nothing that's biologically motivating the fact that these waves exist, right? But a biological pattern would be, for example, those pollen grains, right? That is a biological system. On the surface, you can see behind me, these are patterns that exist on the biological system. So that's how I'm distinguishing the 2. Does anybody want to take a guess?
Hi, Asja, Alex Blumendahl here. Let's say that the bottom right and the top left are physical. That's my guess.
Fantastic. All right, you are the only person to have gotten it right other than my own PI, which is amazing. Do you have a guess as to what they could be potentially?
Oh, I think the bottom right are sort of the sand patterns on the below the water. But the top left, I'm not really sure.
Great, thank you for that. Yeah, so this is what each of these systems is. So as you said, the top left and the bottom right are both physical, and these two are biological. So this top left one is actually the Rayleigh-Benard convection cells. So for those of you that don't know, all you have to do is have a fluid. In this case, I think it has some sort of ferromagnet in it, so you can actually see the patterns. But you just have a differential temperature between the bottom and the top, and this creates these convection cells. The bottom right is, oh, okay, I'll go in order, sorry. The one on the left is actually a diatom. So what you're seeing is the silica shell that the biological system produces as a skeleton on its surface. This is a schematic of bacterial colonies, and as you said, this is petrified sand. Awesome, okay. So the whole point of this slide was to just reiterate the fact that yes, you really can have biological systems that use purely physical principles to form their patterns, have nothing to do with the genetic code necessarily, except for the genetic code forms the raw products with which they're working. This concept isn't new, of course. A lot of people work on this, a lot of people take this approach, and it was first sort of mentioned in On Growth and Form by Darcy Wentworth Thompson. And since this book was published, this idea has grown to argue that complexity in biology arises from, as I've been saying, the genetic code evolving these building blocks and then the building blocks self-assembling. So this is the way that I'm going to approach this problem of patterns on pollen in this talk. Okay, so let's get started with the details. What I have pictured here on the right are 3 different species of pollen. And the whole point of this picture is to show that you get a huge diversity and range of patterns on pollen. So on the left top group of pollen, you have these stripy patterns that come from a houseplant called the peace lily that some of you might have in your house. On the right, these are passion fruit or passiflora, which is the flower that produces the passion fruit pollen grains. And as a physicist, I would argue that these patterns look sort of foamy, but in the literature, they're mostly called reticulate often. And then on the bottom, you have some pollen from sunflowers or daisies, and they're spiky, as you can see. And so the big question when we began this project was, how in the world do these patterns form? Very simple, no one knew, and we just wanted to understand what the physical process was, if there was one, forming these different patterns. Just to give a little bit of motivation of why this question is interesting, biologically it's interesting because these are not the only surfaces in nature to form these types of patterns. They're actually quite common. And they're also common in the sense that they range across a huge diversity of organisms. So each column here represents a different type of organism. We have pollen grains, fungal spores, and insect eggs. And they all come from different kingdoms even, right? So fungal spores are in the kingdom fungi, and insect eggs are of course in animals. And so you get this sort of conserved pattern forming process on spherical surfaces across a huge diversity of different types of organisms. So you can see in the top row, you have stripes. In the middle row, you have spikes, and in the bottom row, you have holes that kind of form this, what people call again, reticulate or foamy pattern. Okay, so happens in a lot of different organisms as well. All right, so pollen specifically, as I said, is very diverse in its patterning on the surface. What I have pictured here is an evolutionary tree, and it represents all of the different species that produce pollen. And in the periphery are just a couple of examples of the types of patterns you can see. I haven't mentioned until now, but pollen actually happens to be the male gametophyte of plants. So it carries the male genetic material in gymnosperms and angiosperms. Those are just the 2 types of groups of plants that produce pollen. Specifically, there are spherical cells that have these surface decorations. So they're single cells and they're spheres. And then the really cool thing here is that each species has a unique pattern. So you can actually identify what species it is based on the pattern that you see on the surface of the pollen grain. And it's so robust that pollen fossils are actually often used for dating in samples. There's no variation within the species, which adds to the fact that they're so robust. And then the last thing that sort of hinted to us as physicists that there could be a physical process going on here is that all species follow the same basic set of biological developmental steps. So somehow, although all of these patterns develop in the same steps, their end pattern looks different. So we wanted to understand how this was possible and what was going on. So in order to do that, we first had to dive into pollen biology and just get a good understanding of what was going on developmentally. So on the top left, I have a schematic of a flower, and the portion of the flower that produces pollen is called an anther, which many of you have probably seen. And inside of the anther, which is schematically pictured here, I have a different screen here. Can you guys see my mouse? Okay. You have a fluid material, and you have between hundreds to thousands to 10,000s, depending on the species, of pollen grains floating around. And they're all developing inside of this fluid material. You start off as a single cell during development, and because this is a sex cell, you're going to go through meiosis. And so you start off with a single cell which has a cytoplasm. The black here is cell membrane, and then this is a transient wall that sort of protects the pollen grain. It goes through its first step of meiosis, or second step, where it produces 4 daughter cells. That's what these 4 circles here represent. And as you can see, they're all encompassed within this outer wall, which is made of cellulose, basically. And then there's a very interesting step that happens next in develop that we're particularly interested in which there is a material that I'm going to call the Primexine that gets deposited from the inside of the cell, of the individual cells, to the surface of the individual cells on top of this cell membrane, but still within this encompassing cellulose wall. At the same time, the cell membrane underneath this Primexine begins to undulate. So that's what was known in the literature when we began this project, some magical process of this Primexine getting deposited and cell membrane underneath it undulating. In the final step of development, all of this material on the surface gets enzymatically digested away, and you get a new material that gets deposited. This new material is called sporopollenin. It's known in the plant world to be a highly, highly chemically and physically robust material, and it encompasses the overall pattern that you see in a mature pollen grain. So in this picture behind me, everything you see here is made of sporopollenin. This is that same species. This is made of sporopollenin. It is deposited in such a way that it mimics whatever this cell membrane undulation was during this particular step of development. Okay. Oh, and one last thing. The reason why this is called Primexine is because this entire surface, this sort of layer, is called the exine. So people sort of postulated from the 80s that Primexine had something to do with being a precursor for the exine. Okay, so this was what was known, but we wanted to understand this particular developmental step in a lot more detail. We wanted to basically answer what is happening with this Primexine, why or how is the cell membrane underneath it undulating, and is it really true that the pattern is established at this particular stage? So before we could be sort of theorists, we had to become experimentalists for about a year because we wanted to actually get a very good understanding of the minute developmental steps within this one particular phase of development. And so our results show essentially this progression from left to right is that development. So the schematic up here is a cross-section of a single pollen cell and describes what's going on essentially. So you have this cytoplasm, you have the cell membrane, on top of it you have that Primexine, and then on top of that you have that encompassing wall. And in the next step of development, you actually start to see a demixing of this Primexine material. So in these actual experimental pictures right here, I've removed this encompassing callus or cellulose wall, so you can actually see what's happening, and you're actually looking at that physical Primexine. And you can see here it starts off smooth, and in the next step of development, you can see that it starts to slowly demix. And then in this third step, you can see that you have a full local demixing into a dense phase, which is labeled in dark purple. and a more fluid phase, which is labeled in light purple here. And so in the schematic, if you look at a cross section of this cell, what you would see is that you have these dense phases that are sitting within these fluid regions. And these dense phases happen to sit within the divots of the cell membrane. So something is happening such that these dense phases are correlated to the undulations of the cell membrane. And then after this, the sporopollenin, or that final material, gets deposited again in the regions between these dense phases and where the cell membrane is sort of coming up like this. And so this is an actual cross-sectional image, unlike these, which are surface images. This is a cross-sectional image where you can see, I've colored again in dark purple these dense phases, and they're sort of sitting in these divots of the cell membrane. Okay, so what we essentially discovered with this particular sequence of images is that the Primexine is demixing, or to be more physically precise, it's phase separating. And that phase separation is what's probably causing the pattern to be templated. You can see this a little bit better if we compare a phase during development where you can see the Primexine material, and then you can look at it in the final form. And what you see is that the sporopollenin is getting deposited in these regions where there's no dense Primexine. So essentially this dense Primexine is creating the negative template for the final form. All right, so hopefully this is clear. It's difficult to match these one-to-one because they're not exactly, this is sort of a foamy pattern, but, and they're two different samples, they're not the same sample, but I hope you guys can see that this is essentially the negative template of this one. So our conclusion from the experimental portion of our study was that the Primexine, which we now know, and I'll talk about a little bit more in the next slide, to be a polysaccharide mixture, locally phase separates into a dense phase and a fluid phase. And another way to look at this is that overall, what you're getting when you go from the not mixed phase to the mixed phase is a phase transition. And you're phase transitioning from a uniform phase to a patterned phase. So this is a little bit confusing language because you have the word phase literally everywhere. So I just want to reiterate this to make sure everybody understands. So a dense phase is essentially a maybe chemically different material that consolidates in a different way than the fluid phase, but it's all still going to be this polysaccharide mixture. But you can argue that maybe you have one type of polysaccharide that's in this dense phase, and then you have another type of polysaccharide that's in this fluid phase. And so just like oil and water phase separate, if you have them, that's what's happening here. You literally just have the phase separation of these different types of phases. And then what's happening in a phase transition lens is you're going from a overall uniform mixed phase to then a patterned demixed phase. Okay, so before I get into how we describe this mathematically, I want to first mention really briefly this Primexine material because I keep saying the name, but I don't really say what it is. And the reason why is because people don't really know what it is, but it's considered a transient plant cell wall. And basically studies done in the 60s showed that it's made of likely a high molecular weight polysaccharide mixture. And the problem is that the features are below the resolution limit of optical microscopes. So it's really difficult to do any staining studies and actually see in detail what this is. But we wanted to kind of get a little bit of an idea, so we sent some samples ourselves to the University of Georgia's Complex Carbohydrate Resource Center and did a monosaccharide and linkages analysis to essentially conclude what are the potential and possible macromolecules that are formed from this. And what we found was that it's likely forming things that are typically found in plant cell walls, which I guess is the conclusion you want to get even if it's not particularly new. But we found that it's creating these hemicelluloses, celluloses, pectins, and then different types of pectins. So something in this material here is producing all of these, or is made of all of these different constituents. Interestingly enough, we sent 2 different species and we found different percentages of these monomers and linkages. So it's likely that they're producing different amounts or even different types of complex carbohydrates with different species. Okay, so yes.
Did this knowledge yield any insight into the phase separation, the mechanistically, or no?
Yeah, so that's actually the very next slide. We were interested if it's even known or possible that these types of constituents can phase separate, if that's what you're asking. Yep. And so we looked into the literature, and yeah, this has been shown before. You do have mixed polysaccharides that do exhibit phase behavior. And some have even been implicated for the properties of cell walls already. Exactly. Okay, so with this sort of under our tool belt, we felt pretty confident about arguing that this was a phase separation process, and we wanted to go ahead and start actually writing down a theory for what was happening. All right, one caveat that I always give, because it's always a question inevitably, is how do you know that you can apply this phase transition to modulated, by the way, just means patterned. It's just a term that's used in the field. How can you apply phase transitions to modulated phases that are going to equilibrium when biological systems are often not in equilibrium? And the way that we argue this is the fact that we're actually creating a material inside of the cell, but then we're not, the cell is, but then that material is deposited onto the surface of the cell where there are little to no metabolic processes. And so we argue that actually it can go to equilibrium because it's external to the cell, not internal. So it's not like all of the stuff that you see with membraneless compartmentalization of organelles that are inside of the cell and that are constantly actively moving and have a lot of energetic changes. This is not the same system. It's going to the external portion of the cell. Okay. So we wanted to use a theory of phase transitions to modulated phases. And for those of you that are unfamiliar with this field, again, it's essentially a way to describe how you go from a uniform, well-mixed phase to a phase-separated patterned phase. It's been used previously in a lot of two-dimensional systems, extensively actually, both from an experimental and a theoretical point of view. So in this bottom left corner, these are images taken from a paper that is describing how diblock copolymers produce these sort of fingerprint-like patterns and also these hexagonal patterns on surfaces in 2 dimensions. You also can see it recently in the literature through 3 different systems. The first is to describe the phase transition process of viral capsid assembly. So it's argued that all of these proteins that are found on the surfaces of viruses actually go through a phase transition process to form the final form. And a very similar theory of phase transitions to modulated phases is applied to this system. You can also see it in systems describing similarly diblock copolymers, but on spheres. And then finally, a really cool application, we work with a lot of people that do this, is for lipid rafts. So these are, this is a fluorescent image of these sort of high density lipid domains found on top of a giant vesicle. Okay, so essentially this theory is well developed already for 2-dimensional systems. It's starting to be developed for patterns that are on spheres, and we kind of were one of the first people to develop it for a pattern that you essentially can imagine as a 2-dimensional pattern that you've now wrapped onto a sphere, so it acts as a shell. Okay, so just to break it down even further, what we're essentially doing is we're trying to model a phase separation that picks a characteristic wavelength, meaning a single wavelength, of the pattern on a spherical surface. Let me caveat this again with the fact that we're looking at a purely phenomenological model. We're not looking for something that describes from first principles how this happens, but rather we just want to do a statistical mechanical approach that can tell us how you go from a no pattern phase to a patterned phase. So the way that you do this in statistical mechanics is you start by writing down a simple Hamiltonian. If that word is foreign to you, just think of a free energy, the energy that you can, that is available to you to do some sort of work in your system. And there are 3 components for our entire system. The components here are drawn. The first one is the Primexine. And so we know that this material has its own free energy. It's doing its own thing, which is phase separating. We also have a second component of our system, which is the membrane. The membrane has an elasticity, it has a surface tension, and it's sort of doing its own thing, and we can model the free energy for what it is doing. And then there's a coupling between the two that's induced by this transient cell wall that I mentioned earlier that holds everything together. So if there was no cell wall here, the Primexine would probably just float off and wouldn't actually interact with the cell membrane. But because it's here, it essentially acts as a type of rigid wall. And so as this process of phase separation is happening within the Primexine, because this is blocking the Primexine from doing anything else, the Primexine has no choice but to push down and buckle the cell membrane underneath it. Okay, so that's the intuition of what's happening physically. And essentially you can model that by just writing down the energy for each of the different components. So before I go into the details of it, I want to give an intuition of why in the world you get patterns instead of just a full phase separation. So what I mean by that is if you imagine that you can somehow have a sphere in which you smear an oil and water mixture on the surface and it's mixed, you would assume, and mathematically this is also what you would get, that you would eventually get the full phase separation and you would have one hemisphere full of oil and the other hemisphere full of water. You wouldn't just purely with this system get patterns like you do for pollen. And so you essentially need a second component of your system to be able to pull out that length scale or that wavelength that actually gives you a pattern. And in this particular system, that happens because there is a membrane that can bend and that you have a callus wall that actually couples your phase separation process to that membrane. All right, so the way that we do this mathematically to break it down is we write down first a Landau-Ginzburg free energy for the Primexine. Again, for those of you that don't know, Landau-Ginzburg free energy is a well-established free energy that was originally written to understand superconductors. But again, it's just a phenomenological model. It takes your field that you are representing, which I'll go into details in the next slide. And it just models how you can go from a mixed state to a phase separated state. The second step is you add your membrane free energy and the coupling between the two. And then you integrate out all the terms that don't have to do with your Primexine and write down a free energy in terms of just this Primexine concentration to find what your patterns are. Okay, so if this doesn't make sense, I'll go into the details in this next slide. Yeah.
Oh, if I may ask a question.
Yeah, please, thank you.
Thanks for clarifying the thing about the oil and water, or why would it not just purely separate and form a pattern? I think a related question that I have is, why is the initial state purely mixed? Like if I think of oil and water, it would be so hard to achieve that state to begin with.
That's an excellent question that I've never got before. If I had to guess, it would be that as the cell is creating these different complex carbohydrates within it, because the cell is still energetically very active, they probably can be in a mixed state. And as they get bled to the surface, they're sort of bled in a mixed state. And then we start to see them phase separate. We know for a fact that it is true, right? Because we do see that it starts in a mixed state. But why it starts in a mixed state biologically, I don't actually have an answer. That would just be my guess, that the vesicles allow for that to happen.
Thanks.
Yep.
So Asja, the membrane, it's bendy, but I guess the intuition is it doesn't want to bend too much. So you don't want depressions that get too deep, and that kind of compromise is what creates the characteristic length. Yep,
that's precisely it. It's exactly the energetic tension between the two. If you have too much of one or too much of the other, you wouldn't get patterns. You would just get a full phase separation. Yep. Okay, so let's go into the details. Since you guys seem to be happily following along. All right, so we'll start with first the Primexine. All right, so psi we're going to treat as a concentration field, and it represents our Primexine. All it represents is the fact that we have a concentration of something, and that concentration happens to be a mixture of two things that want to phase separate. So you can say that you have, a dense phase and a fluid phase, as I mentioned before. Whenever psi is 0, that means you have an equal mixture of the two. And whenever psi is greater than or less than 0, you have a higher concentration of one type versus the other. So all that means is, if you look at this particular graph down here, where we're representing our free energy of the Primexine versus our Primexine material, right here, this is, oh, sorry, let's look at the blue one first. So the blue one is where you don't have a phase separation yet. You have an equally mixed system. So your lowest energy state that you're sitting in is going to just be psi is 0, they're mixed. As soon as you have the phase separation process occur, your graph now looks like this curve, that's orange, and your two different lowest points right here represent for example, the fluid phase versus the dense phase. And so you can see that there is a phase separation of the two because this is the lowest energies that it's sitting at. So that's how the theory works. And then whenever we want to write down our field, we have to write it down in terms of spherical harmonics. The reason why is because spherical harmonics essentially represent the basis states of any function that you want to write down that's on a sphere, right? And so, you know, if you were in 2 dimensions, you would do it in sines and cosines, but if you were on a sphere, you do it in spherical harmonics. Just a quick reminder from my next slide, the spherical harmonics are used a lot in chemistry, right? They represent the different orbitals, but if you look at these little pictures here, they also just represent what the patterns on a sphere can look like. And so what happens whenever you do whenever you break down your function into spherical harmonics, which are these YLMs, you're adding up or you're summing all of the different weights of each individual spherical harmonic. And that's how you can make up a new function that's made of these basis states. Okay, so that's how we represent our actual pattern. It's a summation of the different spherical harmonics weighted individually. And so now all we do is we write down this Landau-Ginzburg equation. And let me break this equation down a little bit. So this is that free energy. Again, it's just describing essentially a Taylor expansion of this particular field. And you have different terms that have physical representations that are different. So this first term is essentially telling you that you're penalizing a change in this field. So that represents interfaces. So you don't want to create too many interfaces, which is why if you just had a phase separation, you would have one entire hemisphere be one material and the other hemisphere be the other material, right? Because you're minimizing that interface between them. So that's what this term represents. This second term is essentially the knob that you twist to get a phase separation. So if you're thinking of, or a phase transition, if you're thinking of ice and water phase transitions, it would be a temperature term. So this is often called a temperature-like term. As you lower the temperature in this graph, you're going to go from this particular curve to this one. So this is just the phase transition knob. And then we won't worry about these for now. These are just higher order terms. Again, like I said, in this Taylor expansion, you could go higher if you wanted to. We just cut it off at the 4th order term to start with the simplest theory that we could. But these coefficients in front of our field represent different combinations of physical parameters. And the way that we've written this currently, as I said at the beginning, this is a phenomenological model. So we don't know what these necessarily represent in our pollen system. They're purely phenomenological parameters, and we don't actually know what they are. We can have a couple of guesses, but we don't actually know what they are. Okay.
Asja?
Yes.
This super naive question, but where are the 1st and 0th order terms?
You don't actually have to add them for several reasons. How do I want to explain it quickly? You typically argue that because of symmetry, you're not going to add those terms. Do you mind if I come back to that just because it's a little bit more detailed?
Totally.
Okay, remind me at the end. Okay, so we now have a model that describes just this phase separation process of just this Primexine. Now we wanted to add the model for the membrane itself. So we didn't actually, just like we didn't invent the Landau-Ginzburg equation, obviously, we didn't create this equation on our own, but we borrowed it from a paper written by Leibler and Andelman in 1987 on ordered and curved mesostructures in membranes and amphiphilic films. But all we do is the same sort of basic template. We take some sort of height field this time for the membrane. And we assume that it can bend and fluctuate away from its original spherical shape. This height field ends up being some sort of baseline plus these fluctuations. So H naught is gonna, you can think of it to be like a radius. And this field U is the height fluctuating away from that baseline. So if you drew a squiggly curve around the actual sphere of the membrane. Again, positive or negative just represents fluctuations above or below the baseline. And again, we're on a spherical surface, so we're representing this in terms of the spherical harmonics as well. As I mentioned, we didn't invent this equation. However, there are different models you can use for this. We happen to use this one. The important thing is that you have a bending rigidity, a surface tension, and of course are representing the radius of the pollen grain within this equation. So you're just making sure that you have a knob that you can tweak that says that your bending rigidity can be increased or decreased and your surface tension can be increased or decreased. And those two parameters are enough for you to create this energetic tension that keeps you from fully phase separating. The coupling I didn't write down because it's details that probably aren't necessary, but we add that coupling and then we do all this sort of massaging of the equation to get it in terms that we want to work with. And so as you can see here, we no longer have a height field. There's no H anywhere in this equation. Instead, we have everything in terms of our psi, which is our representation of the Primexine concentration field. The cool thing that happens mathematically here is that now all of a sudden we have an L naught term come up, and this L naught term is approximately the inverse of the wavelength. R again is the radius of the sphere. And so what this means is that after coupling these two to this, coupling the Primexine to the height field of the cell membrane, you get out a term that tells you that you pick out a characteristic wavelength. And so this is what proves to us that we do get this length scale and we get a pattern and not just the full phase separation. So that's what that looks like mathematically. These are just high order coupling terms, don't have to worry about those. And this is the main portion that we're interested in, where again, you get this characteristic wavelength. And then this term should remind you of that second term from the first equation of the phase separation process. It's just a massaged, newly characterized temperature-like term. So this is that knob that you tweak to get a phase separation process. All right, details aren't super, super important if it's a little bit unclear. What is important is that we now take this giant complicated equation and we start to tweak different parameters in it to explore the phase space and find what are the lowest energy states that nature is going to realize. So the way that we do this is we just stick this in, oh, that'll be the next slide. So let me first clarify. Because this is a summation of, 0 to infinity, just like any spherical harmonic, angular and, momentum terms are, we need to do approximations because we can't go from 0 to infinity. So we're going to assume that L here is equal to L naught, which is a 1-mode approximation. And then we'll go ahead and do a 2-mode approximation where we just let it be L and L plus 1 for simplicity. You can do more. We did do more just to make sure that our lowest energy states were representative, but this is kind of the first order thing to do, is just to do an approximation instead of summing all the way from 0 to infinity, because that's impossible.
So does that mean that L naught is sort of like the possible values for it are kind of quantized, like that according to... Oh wait,
so for L naught, no, sorry. For L naught, they're not. For L, they are, but L naught can be any value.
Right, okay.
Yeah, my bad, sorry.
Asja, on the previous slide, you had L naught being approximately equal to the characteristic wavelength. Why is it approximate?
Good question. How do I...
We can also discuss in the discussion after.
Yeah, I think we'll have to, just because it has to do with the details of how to massage this equation, and I can't think of a quick, simple way of saying why. Yeah, I can't think of a quick, simple way of saying why, but it has to do with the way that this equation is massaged, but we can get back to it.
Okay, I was asking because of what you and Alex were just discussing, I guess, the interpretation for L, and I mean, obviously this term with the difference L minus L naught.
Yeah, so L naught can be any value. It doesn't have to be an integer. It doesn't have to be quantized, but L obviously is, because that is what it is. They're quantum numbers essentially, right? When you're doing spherical harmonics.
I have a question about the higher order interaction terms.
Yeah.
Do they break the azimuthal symmetry?
No, they shouldn't.
I see.
Yeah. Yeah, they shouldn't. Actually, that's actually how you get all these different. So these are Wigner J coefficients that actually have a lot of different symmetry rules that shouldn't. Yeah. Okay. All right, so we tweak these two different parameters to explore what our phase space is. And of course, we're trying to get them in terms of what the actual function on the surface should be. Okay. And this is an example of what it looks like. So we're trying to actually calculate what these different weights are, which are represented here, by finding the lowest energy state of this particular free energy. And so the way that we do that is through simulated annealing. Simulated annealing, unlike things that you might be familiar with, like gradient descent, actually finds the lowest global energy, not local minimas. And so we just created a code that did this to find what the actual lowest energy state was. We confirmed that it really was the lowest energy state by doing gradient descent, and then confirming through diagonalizing our Hessian matrix, for those of you that are interested. And we're able to write down or draw what our phase diagram looked like. And you don't have to go through all that. All right, so this is a simple phase diagram that we ended up coming up with. We did calculate a lot more phases for higher L naught terms, but they're just not included in this particular phase diagram. But let's explore what we found. So we essentially tweaked these two knobs, these two parameters, which are represented on our axes here. And what you can see is that L naught, of course, is this sort of inverse wavelength value. And you can see that as L naught is increased, you end up getting patterns that have smaller and smaller wavelengths. So you go from something like cubes to something like icosahedras. If you look at L naught values that include two modes, so not just a plane mode, which would be like 3, but they include mode 3 and 4, which represents anything in between these two integers, you start to get interesting patterns come out. And the one that I find most interesting is that if you look right where u equals 0 for all of these half integer L naught values, you get stripes. You can notice they're chiral. We do get the degeneracy of chirality, so we get the other-handedness. And I'm happy to discuss why we get chiral stripes and not just plain stripes, and how we could get plain stripes if we wanted to. And the other cool thing is that U ends up being, as probably could have predicted, because we have a third order term that breaks the symmetry, it ends up representing whether or not you have a spike or a hole, which is dependent on whether U is positive or negative. Okay, so we have a phase diagram that can describe a lot of these different patterns. And because the whole point was to model pollen explicitly, our next step then was to try to see if we can match our simulations to real pollen SEM images to our simulations. And so we essentially found this really beautiful database online called PALDAT. It's a database in which palynologists, people who study pollen from all over the world, can upload their images. This screenshot was taken months ago, so at this point it's probably even higher than this many pictures, but they're on the order of tens of thousands of pictures we can skim through. And we, just by eye, tried to fit what could match our simulations. And this is a representation of some of the examples that we found. So obviously they're not perfect matches, but I am happy to say that we felt like this was representative of a lot of the patterns that you can see. So you can see that we get spikes, we get some holes, we get some stripes, and so on and so forth. So I'll let you guys kind of stare at that if there's anything that's confusing or if you have any questions about this part.
You used a word that I can't repeat because it was new to me a moment ago, which had to do with the stripiness, and I assume it had to do with whether the stripes are kind of going down or whether they're kind of going like this.
Chirality, chiral?
Yeah, thanks.
Yeah.
And I kind of see maybe in your examples here that in the kind of orange stuff, which I guess the simulation stuff, you have the chirality, and then the matching electron microscope, they're kind of straight. Yeah, can you please say a little bit more about?
Yeah, so we have a couple of hypotheses. So first of all, as I mentioned at the beginning, we're doing a couple of approximations to the theory that can be fixed on a second round of sort of working on it. The first was we only stopped up to the fourth order term. You could add more order terms. Perhaps that would then end up getting these mixtures of different spherical harmonics that allow you to get something that's a straight line. Or perhaps we could add more modes. So we were curious about that as well, and we did end up adding a couple of more modes to see if you would eventually get stripes that are straight, and we did. We didn't get latitudinal stripes. I always get that wrong. No, these are longitudinal. We didn't get longitudinal stripes, but we did get latitudinal stripes, which I can show you in a slide that comes at the end of the talk. There could be a biological reason as well. Maybe it is really difficult energetically to get longitudinal stripes from the theory. And maybe it works in the pollen system because you end up having, let's say cellulose fibers that end up being particularly stiff and rigid and don't want to bend in that way. So that's also another possibility. That's just a total hypothesis and a hand wavy answer, but could potentially be what's going on.
Nice, thank you. And another question is the graph you had just a moment ago with U and L naught. You don't have to go back to it. I just wanted to invoke it in your head. And then you mentioned that these different patterns each corresponds to different species of pollen. And I don't know if these species are in any way organized on a tree of some sort or something, but does, L naught or U in some way, help or correlate with some kind of organization?
Yeah, we were so hoping that would happen, that we would create this beautiful phase diagram and we'd just be able to impose some sort of tree on it. It did not happen. So no, it's a lot more complicated than that. We can probably get some clues as to what U and L naught could be based on the tree, but it's very, very complicated and will require either a lot of genetic testing and manipulation, or creating a synthetic system where you can tweak certain parameters to be able to actually map out what these phenomenological parameters mean.
Thank you.
But yes, that was our hope that did not happen. Okay, so at this point of the project, although we were quite happy with this progress, we also noticed something kind of interesting, which was as we were combing through the database, only 10% approximately of the patterns that we were looking at actually formed these sort of really beautiful symmetric patterns on the surface that have this one characteristic wavelength. So the rest of them actually looked a lot more, what I would call disordered. That's not really a term, but they sort of looked more foamy like this. And if you wanted to measure out all of the distances between two of these domains, you would find that unlike these patterns, which if you did the same thing and measured, you would get sort of consistent values, here you don't. You get a range of different wavelengths on the surface. And so you're not actually reaching an equilibrium state in 90% of these pollen grains. We didn't, yeah.
So sorry to interrupt.
Oh no, go for it, please, please.
Oh, sort of a naive guess might be that the energy wells are just not as deep once you're way up there in the high harmonics, and it just doesn't care as much exactly which length. Is that?
Yeah, that totally might be the case. Yep. It also could be, we have a couple of different hypotheses that there might be, because biologically it might not matter as well, there might be an influx of some sort of ions that end up cross-linking the patterns prematurely to the actual full evolution of the pattern. And so, yeah, they just don't ever reach the equilibrium state because it takes too long, and they kind of don't even care anyways, maybe. Yep. Regardless, we wanted to be able to simulate at least a proof of concept. So we didn't really do a lot of work on this, but we did do a quick conserved dynamics model with our collaborator, where we just kept the volume of the material constant on the surface and just evolved it with a finite solver to finite volume solver to show that as time progresses in the simulation, you do end up getting something that looks foamy. So these on the right are our actual simulation results, and where the range of wavelengths is getting smaller and smaller, larger and larger, sorry. And these are some of the patterns to the left that we think could be representative of this particular stage of evolution as the phase separation process is occurring, and then something is stopping it from going to completion. Whether or not it just takes too long and it doesn't care, whether or not there's something being introduced to the system, we don't know the answer, but it does seem to be a kinetic arrest process, which just means something stops it from going to completion. Okay, so at this point, we essentially felt pretty confident in saying that all patterns can be characterized in either these equilibrium states. Again, only 10% of them actually get to these really beautiful symmetric patterns. And then the rest of them can be described by these kinetically arrested states that end up looking a little bit more foam-like. And again, we didn't really do a lot of characterization here, just more of a proof of concept. Okay, so there's a lot of different things that we think that we can do with this theory now. I'll just give two different examples, but I won't really go into a lot of detail. I also have literally no clue what time it is. Okay, I'm pretty much there. So one example is you can use this to do an evolutionary analysis. So one question that hasn't come up is why in the world pollen forms these patterns? The quick answer is there's no consensus. There's probably a lot of different functions. And no one really has pinpointed why you would get one pattern versus another if you're trying to achieve a particular function. But these evolutionary analyses are a little bit difficult to do at times because the pattern definitions are completely arbitrary. Palynologists have essentially said, biologically, these look similar, so we'll call them this. But that probably has nothing to do with the actual, as Alex mentioned earlier, phase diagram. There's no actual mapping between the two. So we argue that you could essentially base your trait analysis on this pattern formation mechanism to try to actually say, okay, this pattern is similar to this one because it has similar L0 values or U values or something like that. So these are all those terms that currently don't mean a lot mathematically, and if you just turn them into some sort of pattern definition based on whether or not they're at equilibrium or what their wavelength is, you might be able to find answers as to why or how certain patterns evolved from other ones. And then the second thing we're interested in using this for is to create synthetic pollen. We have a collaborator that's done this recently that we worked with, where they created pollen-like liquid crystal elastomer particles. They took this mixture and a similar process of a phase separation and then coupled to some sort of energetically limiting length scale allowed them to get these patterned particles that are elastomers. Okay, so thank you very much for listening and asking questions, and I'm happy to take any more if time permits.